Good morning, everybody. So we'll give people a minute or two just to kind of start rolling in. Good morning. Looks like we still have some people rolling in, so we'll give it another minute or two. All righty. So good morning, everybody. Thanks for hopping on this morning. We're really excited for today's webinar. Before we get started, we want to take a moment just to recognize all of those in Florida, those that are affected by the hurricane, those that have passed away. Our hearts and prayers really go out to all of those that have been affected, their families, and we hope that everyone can remain safe and are able to recover quickly from the events that they've been having down in Florida. So I would like to welcome you all to the second session of a series of isotope days featuring cutting-edge science facilitated by stable isotopes. Today's session focuses on NMR and other related techniques and technologies. My name is Kelly Andrade, and I'm going to be your host for today's event. So there's a couple goals we hope to achieve in today's webinar. I hope that we're all able to explore the cutting-edge applications of stable isotopes, learn something new from the scientists in the NMR field, the products that CIL offers, and the role that we play in the research field, that you all are able to strengthen your understanding of these products in the area, and most importantly, that everyone's able to stay engaged and have fun throughout. In case you missed it, we also had our mass spec portion of Isotope Day last week, and next Thursday we will have the environmental session as well. Most of the recordings will be posted on our website along with today's session in the coming weeks. Our agenda today is outlined here, and following this brief introduction, we have a wonderful set of panelists today who are going to speak on a variety of different scientific applications, approaches, all utilizing stable isotopes and NMR, as well as other related techniques, and how they use this all to help answer their research questions. Each talk is going to be about 15 minutes with 3 to 5 minutes of Q&A afterwards. Again, as I mentioned, most will be recorded and disseminated after the fact. And if you have any questions throughout the duration of the presentations, please utilize the Q&A box to submit your questions for the Q&A session. And before we get started, we have a brief 3-question poll, which will help us get an understanding of who is in the audience today, what your areas of research are, and the application interest that you have. So I'll give people about a minute to answer the survey that should be on your screen. Alrighty, I'll give like another 20 seconds or so. Alrighty, and another like 10 seconds just to finalize the poll that you guys have on your screen. Alrighty, so we can go ahead and close out the poll now. And without further ado, we can jump into our scientific talks. So first up, we have Dr. Andrew Hinck, professor and deputy chair at University of Pittsburgh. Dr. Hinck received his PhD from the University of Wisconsin-Madison, where he was trained as a biochemist in the lab of Dr. John Markley. In 1994, Dr. Hinck joined Dr. Dennis Torchia's lab at the NIH. There, he collaborated with the Roberts and Sporn group. Oh, my apologies. There he collaborated with the Robertson Sporn Group at the National Cancer Institute to determine the structure of TGFB1, labeling it with 15N and 13C by expression in mammalian cells and utilizing NMR spectroscopy. In 97, he moved to the University of Texas, where he began building up his lab and making inroads toward understanding molecular mechanisms of receptor complex assembly in the TGFB superfamily. In 2015, Dr. Hinck moved his lab to the University of Pittsburgh, where he continues to apply NMR to study structure-function relationships of difficult-to-produce TGFB family proteins produced in mammalian cells. His talk today is titled Synthesis of 13C methyl-labeled amino acids and their incorporation into proteins in mammalian cells. And without further ado, Andy, the floor is yours.
Okay. Well, thanks, Kelly, for the invitation, and I'm excited to present the work we've done in terms of producing proteins in mammalian cells that are labeled with C13. It's as Kelly mentioned in my bio, I actually have been trying to produce proteins in mammalian cells for NMR for some time, and I'm excited to tell you about these most recent developments. So just a little bit of background. I think most people in the audience know that uniform labeling with N15 and C13 and full assignment of the proton, carbon, nitrogen signals has been highly impactful for studying proteins at the molecular level using NMR, particularly very strong for studying smaller size proteins. You can go up to about 25 kilodaltons with just N15, C13 labeling, although beyond that, it begins to become challenging due to rapid T2 relaxation that limits application to larger proteins. You can extend that by deuterating the proteins and using sensitivity enhancement approaches to improve the line widths and extend the application of these methods. Here on the right side of the slide, I show this for 40 kilodalton receptor, the TGF beta family that we studied. We get pretty, this protein is deuterated and N15 labeled, and we're using TROSY techniques. We get pretty nicely resolved proton carbon correlation spectra and pretty good sensitivity on the spectra to enable assignments across the peptide bond. Although you can see for the relevant cross peaks that give the correlations to enable the assignments, they're starting to get a bit weak at this molecular size, even with deuteration and even with sensitivity enhancement. So while, again, this technique's been very powerful, it does have limitations in terms of the sizes of proteins to which it can be applied. Another limitation of I would say, aside from molecular weight limitations of uniform labeling with N15 and carbon, is that it can only be applied to proteins that can be labeled that way, so uniformly. So that's largely relegates this to being used for proteins that can be expressed in either bacteria or yeast. Now, to extend the methodology, Louis Kay's group made some really important advances in this area almost 25 years ago, where they showed that if you look at the side chain methyls, you can observe NMR signals in quite a bit larger proteins with pretty good sensitivity and resolution. And this is due to the fact that the side chain methyls have favorable T2 relaxation properties due to picosecond rotation around the methyl bond axis. You also have increased signal intensity due to the chemical equivalence of the methyl protons. You have insensitivity to pH, which for a lot of larger, more complicated proteins is necessary. A lot of proteins tend to be more stable in the pH 7 to 8 range, so you don't have problems with loss of signal due to solvent exchange. And most importantly, I would say, is Lewis's lab, but then other labs, Jerome Bospevet's lab in Grenoble, developed methods for enable robust labeling of methyl groups in proteins based on incorporation of simple C13 based metabolic precursors, such as alpha keto isovalerate to give labeled methyls of valine and leucine, or alpha keto butyrate to give methyl labeling at the delta position of isoleucine. Bospevet's group showed that if you go a few steps earlier in the metabolic pathway and then label with C13 acetolactate, you can also get labeling of valine and leucine, although one advantage of doing it that way is that the labeling can be stereospecific. So all these approaches are very powerful, I would say well developed and really extensively applied. And I think really has transformed applications of NMR to larger and more complex proteins than could be studied with just carbon and nitrogen labeling alone based on E. coli expression. But the issue is that while the NMR technology is pretty well developed, it's unfortunately really only applicable to proteins that can be expressed in native form in bacteria. A lot of, on the other hand, a lot of larger human proteins, such as highly disulfide bonded receptors, which is a big focus in my lab, or membrane proteins such as GPCRs, they cannot be produced in a natively folded form in bacteria. You can think about using yeast to make some of these proteins, and that certainly is possible for some of these, but I would say not all. For example, a lot of the GPCRs, well, there's been a few examples of successfully producing homogeneous GPCRs for structural work in yeast. The number of examples are actually pretty limited, and actually most GPCRs cannot be readily made in using yeast expression. There's also some disadvantages of the methyl labeling in yeast. The labeling efficiency is not as high, and the extent of labeling that you can get compared to bacteria is not as high. Now, in terms of going to, if you want to think about studying some larger, more complicated proteins that, and neither bacteria nor yeast is possible, you can start to think about using insect cells or mammalian cells. These cells are the cell types that normally produce these complicated proteins, and they have a lot of chaperones. They have sophisticated secretion systems. They're really made to produce these proteins in a native form. The challenge, though, from an NMR's perspective is you cannot simply use metabolic precursors in insect cells or mammalian cells for producing methyl-labeled proteins, because those cells lack the requisite biosynthetic machinery. So you have to think about other approaches if you want to do methyl labeling for proteins produced in insect cells or mammalian cells. And one attractive approach is using direct incorporation of C13 labeled amino acids. But one of the challenges with that, I would say, is the lack of low-cost synthetic routes to the common methyl-bearing amino acids substituted with carbon 13 at the methyl position. So that's where we were trying to make some impact. And so what we did was we collaborated with two chemists here at the University of Pittsburgh, Matteo Borgini and his advisor, Peter Whipp. And Peter and Matteo developed a scheme that was based on to enable synthesis of two, so far, C13 methyl labeled amino acids very efficiently and at pretty low cost, as I'll explain in a moment. So basically, I have done some chemical synthesis, but I am not a synthetic chemist. So this was really the work of Matteo and Peter. But basically what they did is they basically start with alanine, L-alanine. They add a protecting group on the N terminus and a what they call directing group on the C terminus, and then they use that to then essentially build up the side chain. And the C13 that's going to give rise to the C13 methyl is introduced in the form of C13 iodomethane, which is a relatively low cost reagent. And so basically using this approach, they were able to synthesize isoleucine, which notably has two chiral centers, one at the alpha position, but one at the beta position as well. So this is a stereospecific synthesis. This synthesis is possible in terms of achieving high stereoselectivity at the beta position due to this directing group that's attached on the C terminus. So that basically coordinates metal, and that leads to high specificity for addition of the methyl group at the beta position. And then once the compound's been synthesized, you have to be careful about the deep protection to not erode the stereospecificity. In any case, this amino acid, C13 isoleucine, labeled at the gamma-2 position, was successfully synthesized in good yield and again at low cost. Peter and Matteo also used a similar approach to synthesize C13 valine that was labeled at both of gamma-1 and gamma-2 positions. Again, this was done very efficiently and also with low cost. This is a less challenging synthesis because in this case, there's just the one stereo center at the alpha position. So these amino acids were then incorporated into mammalian, incorporated into some of the receptors that we study in my lab. These are relatively not very large. They're only about 20 kD, but they do have multiple disulfide bonds. And you can express these in E. coli, but it's actually difficult to refold them and recover natively folded receptor. So they have either 3 or 4 disulfide bonds. We used 2 of them as model systems, one called VG-ZPC and another one called R3-like. We produce the proteins in suspension cultured HEC293 freestyle cells, and we use high clone trans-FX medium. So this is commercial medium without amino acids. And then we add to that all of the amino acids that we don't want to labeled in unlabeled form, and then along with the synthesized amino acid with the C13 methyl label. And the medium that we reconstructed using this procedure is not quite as good as full commercial medium for these HEC293 freestyle cells, but its yield is about half of full commercial medium for these cells. So it's not perfect medium for protein production on these cells, but it does work reasonably well. This shows in this table here, it shows the amounts of the methyl iodide that we used in our syntheses and the amount of the corresponding amino acid that we were able to obtain. So with about 2 grams, either 1 or 2 grams of C13 methyl iodide, which is relatively inexpensive, $200 or $300, you can get between about 100 and 200 milligrams of the corresponding amino acid. And then with those amounts of amino acids that's shown here below, you can produce, using relatively small culture volumes, on the order of 1 milligram of protein. So with just $200 or $300 worth of methyl iodide, you can get to about 1 milligram of protein. And we're looking at the methyl signals, which is very sensitive. This is actually, these amounts of protein are actually enough for 7 or 8 NMR samples. So it's really only about $30 or so per NMR sample. It's actually pretty low cost. So that's how we produce the proteins, and then we, of course, examine them to look at the labeling in terms of its efficiency and whether it's what we expect for the amino acids that were incorporated. So this is for BG-ZPC. This protein has 5 methionines and 13 valines. You can see it in the amino acid sequence here. And this is exactly what we see in the spectrum. This is the isoleucine methionine labeled spectrum. We see the 5 isoleucine signals, or I'm sorry, the 6 isoleucine signals and the 5 methionine. These are the 13 valine signals. In this case, there's 26 signals because it was both gamma 1 and gamma 2 labeled, and again, the 5 methionine signals. So this looks really exactly as expected. The signals are exactly where we expect in terms of their chemical shifts, and then there's the exact number of signals that we expect. And then for R3-like, it also looked good. So we made an isoleucine methionine labeled sample, and we see the 4 expected isoleucine signals and the 1 methionine. And then, so we wanted to know what was the labeling efficiency. Are these amino acids being incorporated with high efficiency? And so what we did was we recorded the spectrum of the same receptor, but with natural abundance C13. And then we compared the signal intensities of the either the valine or the isoleucine relative to methionine. Methionine is known to be very well incorporated with 100% efficiency. So by comparing these ratios with the protein produced in HEC293 cells or in the natural abundance sample, we can determine whether the incorporation efficiency was high or not. And as far as we can tell statistically, it looks like the amino acids are being incorporated with near 100% efficiency. We also wanted to know about the line widths. The one thing about this that you need to keep in mind is that the protein that we're producing in mammalian cells is not deuterated. So we incorporate the C13 methyl labeled amino acid, but it's not possible to deuterate to any significant extent in mammalian cells. The cells, unlike bacteria, the mammalian cells are not as tolerant to high D2O concentrations in the medium. And so we actually expected the line widths of these methyl signals labeled in this way to be broader than you would get from normal isoleucine, leucine, valine labeling from metabolic precursors for protein produced in bacteria with background deuteration. So we actually then, what we did was then compare the line widths in our mammalian produced methyl signals of valine versus the same signals, but in a sample that we produced and managed to refold, not very well. That's why the spectrum looks a little scraggly, but we were able to produce a small amount of native protein by bacterial expression and refolding and purification. We compared the valine methyl carbon line widths in this ILD bacterially produced deuterated sample relative to the same methyl signals in the mammalian. And here's the plot of the carbon line widths, and you can see that relative to valine or relative to methionine in the HEC293 mammalian produced protein where there's no deuteration, the valine line widths are indeed larger than the methionine line widths. Whereas in the E. coli protein, where we have full background deuteration, you can see the valine line widths are more comparable to the methionine line widths. So while incorporation of C13 labeled amino acids using mammalian expression systems seems to be very high, the line width, however, is not as narrow what you can achieve with methyl labeling in bacteria with background deuteration. So that is one limitation of the method. So these are my conclusions now. We've developed an efficient synthetic route to C13 labeled methyl labeled amino acids, at least valine, gamma 1, gamma 2 labeled, and gamma 2 labeled isoleucine. We show high incorporation efficiency using mammalian cell expression. We see little to no scrambling, and we can produce these samples in quite a cost-efficient manner. It's pretty simple to do this, actually. It's actually not technically that challenging. And we think that this can enable studies of larger, more complex human proteins than would be possible using just either bacterial expression or yeast expression. I did point out the limitation of the method in terms of the broader line widths due to the lack of background deuteration. I think that's an area for future investigation. I think one possible approach there is to modify the syntheses to install deuterons at positions directly adjacent to the C13 to minimize dipolar relaxation. Those nearby protons are going to be the ones that most strongly drive relaxation, and replacing them with deuterons should reduce that. And I should say my group is not the only group doing this. Hari Arthanari at Harvard has also been playing around with these types of approaches, and they've also made similar observations to us, and are also working to do exactly what I just described in terms of installing the deuterons to improve the langlets. That's what I have for my presentation. Thank you for your attention. I just want to acknowledge very quickly the people that did the work. This work is primarily done by a postdoc in my lab, Łukasz Witeska, and Cynthia Hinck, my wife, who helped with a lot of the protein expression and purification. And these are our chemists who we collaborated with, Matteo Borgini and Peter Whiff in the chemistry department at the University of Pittsburgh. Thank you again. Happy to take any questions.
Wonderful presentation, Andy. Thank you so much. It's very exciting to see the work of methyl labeling being worked on in mammalian cells. Unfortunately, we don't have time to take the answers live for the Q&A, but we do have them in the Q&A box. If you're able to type the answers in the chat, that would be greatly appreciated.
Okay, thank you.